Welcome to MycoTalks, hosted by the MRC Center for Medical Mycology at the University of Exeter. My name is Judith Berman, a professor at Tel Aviv University in Israel, and I'm one of the members of the organizing committee for this talk series. The co-chair today is Professor Axel Brakhage from the MRC Center at Exeter. Today, our speakers are Professor Axel Brakhage from the Hans Noll Institute in Germany and Ashraf Ibrahim from the Lundquist Institute in the US. MycoTalks are organized by the committee shown at the bottom of this slide with valuable assistance in running the event from the MRC Center staff. MRC Talks are held at 1600 UK time on the last Thursday of every month. They were started by MRC Center back in July 2020 to bring together the medical mycology community to hear about great science during the lack of face-to-face conferences due to the COVID-19 pandemic. Even though we hopefully are now emerging from the pandemic, MycoTalks attract an audience of around 300 scientists around the world, with many more viewings of the recorded talk later on YouTube. Apart from great science, the philosophy of MycoTalks is to promote diversity in our field in terms of career stage, gender, geographical location, and research field. And the committee is always happy to receive suggestions for speakers for our future program. The format is two 30-minute talks on different aspects of medical mycology, followed by a 30-minute question and answer session at the end. To ask a question, please post it in the Q&A tab, which will be seen only by the moderators. It is important to state the name of the speaker you wish to address. The chairs will alternate the questions between the speakers and may choose to amalgamate questions that cover a similar topic. However, even if we run out of time, all questions posted to the speakers on the Q&A will be sent to the speakers after the event. The upcoming MycoTalks are listed on our website and you must register for each session. Please note that the times of the talks are at 1600 UK time, so especially in spring and autumn, check how this might be affected by changes in daylight saving times change status. We would like to draw your attention to the Medical Mycology Trainee Seminar Series, which is also held on a monthly basis and is a great opportunity for early career scientists to give a talk. So now I would like to introduce Axel Brakhage, who will be giving our first talk. Professor Brakhage holds a full professorship in the Friedrich Schiller University in Jena and is also the head of department and director of the Leibniz Institute for Natural Product Research and Infection Biology in Jena, Germany. He is the spokesperson of the Interdisciplinary Consortium Infect Control of the Cluster of Excellence, Balance of the Microverse, and the Collaborative Research Center Transregio on Human Pathogenic Fungi, FungiNet. He serves the scientific community in several positions as editors of journals like eLife and MicroLife, and as vice president of the Deutsche Forschungsgemeinschaft, and as senator of the German National Academy of Sciences, Leopoldina. His research is devoted to two areas: infection biology of human pathogenic fungi with a focus on Aspergillus Fumigatos, and how microbial communities are structured and affected by natural products and novel anti-infectives. I remind you again to post questions in the Q&A tab with the name of the speaker that they are for. And now please, Axel, could you turn on your video and start sharing your talk?
Okay. Yes. Okay. First of all, Judy, thank you very much for giving the opportunity to speak here and for the really kind introduction. I would like to speak about two aspects of my research, and I come back to that in a minute. First, I would like to speak a bit about Aspergillus Fumigators. Aspergillus Fumigators is a fungus that causes life-threatening infections. Sorry, I just have to switch on the laser pointer. And you can see the fungus here growing on an agar plate, and this gray-green color from the colonies results from a certain type of melanin, which I will speak about a bit later. And this gives the gray-green color. And by the way, it also is the name-giving compound because fumigatus comes from the Latin word fumus, the smoke. And here you can see the conidia spores produced by Conidiophores Aspergillus fumigators. And it's estimated that every human inhales hundreds of spores every day because you can find these spores everywhere on Earth. And in particular in immunocompromised patients, it can lead to serious infections in the lung, and then the fungus can spread systemically. And of course, this is really life-threatening. And it's estimated that we have about 300,000 cases of the disease called invasive aspergillosis, and which is extremely severe. Now, what we also see is more and more that we have co-infections by Aspergillus fumigatus in viral infections. And there is now several reports showing that in COVID-19 patients, there is sometimes we observe associated invasive aspergillosis. And this, of course, is very dangerous because of the severe infections caused by Aspergillus fumigatus. In addition, this was also previously reported as associated, for example, with influenza. Now, the problem is with human pathogenic fungi, it is really now a significant problem. And there are several reasons why this is the case. First of all, the diagnosis is often difficult, in particular for Aspergillus fumigatus, and often too late. Therapy is, at least for fumigatus, often ineffective, and there's only few limited therapeutic options. In fact, there's only 3, maybe 4. It depends how you order these compounds, 4 different compounds that can be used clinically. And we even observe for some of these compounds resistance, and also some of these compounds are rather toxic. And also what we have to consider, predisposition cannot be prevented, because many patients that are susceptible for an infection by Aspergillus fumigatus are the result of modern medicine. Immunocompromised patients, cancer patients that are treated, patients of transplant medicine. And all this together leads to the disappointing fact that the mortality of these diseases is extremely high, between 25 to 90%. It is higher. In fact, invasive aspergillosis concerning mortality is one of the diseases with the highest mortality in the clinics, and much higher than for any other disease. And this significant problem has also been realized by the WHO, and there was the first meeting of an expert group on identifying priority fungal pathogens. Now, when we come to the interaction of Aspergillus fumigatus with the immune system, then it's the following path. We inhale conidia, as I said, they are everywhere. And then in the lung, they are confronted with different cell types. On the one side, we have the alveolar epithelia cells, and I think the importance of alveolar epithelia cells for defense against all type of pathogens is underestimated. In addition, and this is what we call the first line of defense against Aspergillus fumigatus, are neutrophils, polymorphonuclear granulocytes. And also to this first line of defense belong macrophages. And macrophages phagocytose, also like PMNs, and then they try to kill these pathogens and also, of course, present antigens. Now, when you think in general terms about the interaction, the complex interaction of pathogenic fungi with humans, we have to distinguish 2 major rules. One is, of course, we and also other animals like mice, which we used in experiments, we try to defend against these fungal pathogens. On the other side, fungi, in particular the pathogenic ones, there is a counter response. They have developed strategies and they have physiological characteristics helping them to evade the defense of the immune system. This is what we call immune evasion. Today, I would like to speak about two aspects of research in my laboratory and to cover both aspects I've just discussed. First of all, immune evasion. And we have been working for quite some time on the interference of Aspergillus Fumigators with the maturation of phagosomes. Now, when you think about a conidium, then what we see is that the surface molecules are in first contact with the immune system. And when you look at this spore as such a conidium, of course, you have a lot of proteins. And we did a lot of proteomics on surface proteomes, etc. And also we identified, and also other groups, for instance, the group of Jean-Paul Lager, the ROD-A, we identified several proteins that are really important for immune evasion. However, today, I would like to one of my favorite molecules that makes this gray-green color, as I said, and this is a melanin. And it's not just a melanin. Melanins are quite complex. They have different structures. This is a distinct type of melanin, which we call dihydroxynaphthalene melanin. What is this complicated name? Early on in my lab, we contributed to the discovery of the melanin biosynthetic pathway and also identified the gene cluster encoding the enzymes of this pathway. And it's a very simple pathway for chemists. It's a polyketide pathway, and the name-giving compound is this very, very simple dibenzoic acid compound, which is dihydroxynaphthalene. And this polymerizes to this DHN melanin, and nobody really knows the final structure. It's rather complex. Now, the initial enzyme leading to this biosynthetic pathway is a polyketide synthase, which we named PKSP. Now, when we delete this gene, you end up with white like snow conidia. And when you compare these white conidia with wild-type conidia, which are gray-green, in a mouse infection model or other models, cell models, you find reduced virulence. And this told us that melanin is really important for virulence. And during this work, we also discovered that it has several effects. It has even multifaceted effects. But I would like to speak about one major effect that we have been working on for quite some time. So I would like to address the question, how does this melanin contribute to virulence? And I would like to remind you, and we come to alveolar macrophages that you can see here, and what they do, and all of you know that, they recognize conidia, we labeled them here with DS red. Then they phagocytose them, and then they process them, what we call intracellularly. And what that means is they put them in phagosomes, and then they try to kill them in these phagosomes. And what we found very early on is that this type of DHN melanin somehow prevents the formation of mature phagolysosomes. And just to give you one example how we carry out such type of experiments. So we isolated conidia wild type and these white PKSP conidia, we labeled them with chalcofluor white. Then they are taken up, phagocytosed by macrophages that you can see here. And then we asked the question, how many of these conidia end up in an acidic environment that you can nicely see here, labeled with lysotracker. This is a phagolysosome. And when you see that already here, from these white conidia, nearly all of them end up in this acidified compartment. And this is very rarely the case when you have melanized conidia. And then, of course, you can quantify that, and you can ask how many of these conidia end up in acidified compartments. And this, with all cells, we checked that, we found the same result. And that means, or nearly with all cells, I must say, for instance, the macrophages, about 20% or 30% in this case of wild-type conidia ended up in such acidified environments. But when you had no melanin, it was about 80%. And this told us there must be a general mechanism of melanin on the maturation of phagolysosomes. And then we ended up with this model when you have non-melanized conidium, and this, I guess, is true for many pathogens. They are phagocytosed, they are recognized, and then the phagosomes fuse with lysosomes, and then they generate a hostile environment in this phagolysosome, for instance, reduced pH and other things, and thereby they kill the pathogen. However, when you have melanin there, this only happens very rarely. It is delayed, and the pH is not really decreasing, and this allows conidia to survive in these phagosomes, and in the macrophages for some time, and they gain time to further develop. Now, the question, of course, which I would like to discuss mainly today is, what is now the mechanism? That how can DHN melanin interfere with macrophages? And to solve this question, we joined forces with two really excellent scientists and their groups. On the one side is Agostinho Carvalho in Braga in Portugal, and Georgios Chamelos on Crete in Greece. And I would like just to show you one further experiment, which explains a bit our conceptual frame, how we further developed. What we did in this experiment is we used one macrophage cell really, and this macrophage phagocytosed conidia, both PKSP conidia, so white conidia, which we labeled with chalcofluor white, so they have a blue fluorescence, and wild type conidia, which we labeled with FITS, they're green. And then all these conidia were in one macrophage. And then we asked the question, what about acidification? And here's the experiment: blue, white conidia, FITS labeled wild type conidia, and then labeling with lysotrack, and this tells you which conidium is in an acidified compartment. And as you can see, this is only true for this white conidia, for this one here, PKSP mutant conidia. And here is the overlay. Here you can see the wild type conidia, and there are nicely these white conidia in these acidified compartments. And this told us that this must be a process within a phagosome. So the question is, how can it happen that a melanized conidium containing phagosome, for instance here, can influence the maturation of this phagosome? For instance, by inhibiting the assembly of VATPase. And when we were further carrying out research, and for this also Scott Filler from Los Angeles was important for many discussions and help, we bumped in lipid rafts. And I just would like to remind you what that is. Lipid rafts were described in the 90s, in particular by Kai Simmons' group. And when you think about a membrane is just not uniform. What you find within these membranes is these so-called lipid rafts. And they're enriched, for instance, in cholesterol, in particular in sphingolipids. But also there's specific proteins in there, like chaperones, which apparently keep them together, but it's not really entirely clear all this. So what we then did is we stained for lipid rafts in these phagolysosomal membranes. The following experiment, conidia, and I maybe explained it with this white PKSP conidia. You can see them here in macrophages, and when you stain now the membrane of the phagosome, and the phagolysosome it will be at the end, it's a nice staining for Philippines. So they contain lipid rafts. And when you compare that with wild-type conidia, you don't see anything. So wild-type conidia are in a phagolysosome or phagosome that does not contain many lipid rafts. And of course, this was very interesting, and we concluded that lipid raft microdomains are reduced in membranes of wild-type conidia containing phagolysosomes. And then the next question is, there's also proteins that are important for it. And I don't go into all details, but I would like to concentrate on two proteins that are very interesting called Flotyline 1 and 2. And we did some initial experiments, and we had the idea that Flotyline 1 is more important than Flotyline 2 in this respect. It's believed they produce heterodimers, but also they can act as monomers. It's a bit unclear. And then we did the same experiment and stained with antibodies against Flotyline. And what you can see here, again, staining now both conidia with chalcofluor white in blue, then they are taken up by macrophages. And then if you concentrate on these PKSP conidia, you can see this is staining, by the way, with cholera toxin, which stains sphingolipids, so also lipid rafts. And here is Flotyline staining. And you can nicely see there is Flotyline-1, it's an antibody against Flotyline-1 in the phagolysosomal membrane. You don't see that with wild-type melanized conidia. There is no Flotyline-1, there is, or at least very few, there's very few staining of sphingolipids. So this further supports something is wrong in these membranes. And then we got a knockout mouse model, and these mice, they have a deletion of both Flotylines 1 and 2. And then we isolated bone marrow-derived macrophages from these mice. And then, of course, we asked the question, how many of the conidia end up in acidified phagolysosomes? This is with wild-type conidia. This is what we would expect, because they inhibit anyway the generation of Flotyline lipid raft membranes in phagosomes. But when we had now the PKSP conidia, it was reduced. And this told us that these Flotylines and lipid rafts are important for the maturation of a functional phagolysosome. And then we did proteome analysis on isolated phagosomes. And for that, we established an isolation procedure which was just recently improved by Marie Goldmann and Franziska Schmidt from my laboratory, published in STAR Protocols. And what we did is then we did a dual proteome analysis of conidia containing phagosomes. We analyzed both the proteome of the host and also the proteome of Aspergillus Fumigators. I don't go into the details, but I just would like to concentrate on the top 10 enriched proteins in PKSP conidia containing phagolysosomes. And if we just concentrate on this boxed proteins, this all is subunits of the VATPase. And this is important to pump protons in the lumen. And this result tells us that wild-type conidia can have somehow reduced VATPase in the phagolysosomal membrane compared to PKSP conidia. And so we hypothesized from this data that DHN melanin somehow prevents the assembly of the VATPase. Just to remind you, the null subunit of VATPase is embedded in the membrane. And for a functional VATPase, the cytoplasmic V1 subunit has to be assembled, and then it can pump protons into the phagolysosomal lumen. And just one experiment, we labeled then again white PKSP conidia with calcofluor white, wild-type conidia with FITs, and then in the cell we labeled the phagolysosomes with an antibody against VATPase. And what you see here, this conidium has far more in the phagolysosome, far more VATPase than in a phagolysosome only with which contains the wild-type conidium. Okay, the next question, of course, to have this cause-effect relationships was, what about the VATPase? The assembly of the VATPase, is it dependent on lipid rafts? And there were already reports that, in fact, that this is the case, but there were no reports showing, as far as I remember, that it's also dependent on Flotyline-dependent lipid rafts. And for that, we went back to our mouse model. And first, I show you data just on bone marrow-derived macrophages from wild-type mice. And again, we labeled with an antibody against the V1 subunit of VATPase. And what you can see when you have PKSP mutant conidia, stronger staining, more fluorescence than compared to wild-type conidia. But the important experiment is, of course, when you compare now the Flotyline knockout mouse with the wild-type mouse. And then again, assembly of VATPase, when you quantify that, and you have it here, wild-type conidia, there's not much difference. This is what we would expect. But when you have now PKSP conidia, usually there's a nice assembly of VATPase. Here it's something like 30%, it doesn't matter. But when you have no Flotyline-dependent lipid rafts, it's heavily reduced. This tells us we need Flotyline-dependent lipid rafts for the full maturation, for the full assembly of VATPases, and that this is reduced by wild-type conidia, basically by melanized wild-type conidia. Now, and last point I would like to discuss on this project is then we looked for the Flotyline 1 gene in humans, and we have in humans one gene for Flotyline 1 and one for Flotyline 2. In mice is the same. And then the group of Agostinho Carvalho screened single nucleotide polymorphisms in this gene. And they found one, which is shown here as a T2C exchange. This is the name, it's on chromosome 6. And with this Flotyline 1 SNP, we found an association with invasive aspergillosis. So for that, Agostinho's group analyzed hematologic patients undergoing allogenic hematopoietic stem cell transplantation. They had about 79 cases of probable proven aspergillosis and 244 uninfected controls. And what they found is that the donors of the stem cell transplantation, when these donors had this SNP, there was an increased risk for the recipients after receiving this stem cell transplantation for invasive aspergillosis. And this increased to 38.5%. And this is the probability score, which you can see here. So if you have donors that have wild type situation for Flotyline, then it's about in this range. When you have an heterozygous situation, then it's already increasing. And when it's homozygous for this SNP, then it's at 38.5%. And of course, this is, I think, a nice further strong hint that Flotyline-derived microdomains, lipid grafts, are very important for immunity, for the defense against infections by Aspergillus Fumigators. At the end, I would like to show you to this project our recent model, and this is based also on many data of Georgis Pramilos and Agostinho Carvalho. So when we have here the membrane, and we have non-melanized conidia, they are taken up, they are then in the lumen, and what is happening is that there is the formation of lipid raft that you can see here with flotylines. This is a bit speculative, we don't know whether it's really heterodimer. And then this is a platform very important for assembly of VATPAs, also NADPH oxidase. I didn't show you any data, and most likely also for many receptors. Now, when you have melanized wild-type conidia, then there is no formation, or at least reduced formation of these lipid rafts. And as a consequence, there is reduced assembly of VATPAs, NADPH oxidase, and so on. What could be the molecular cause? And there is very good data from Georgios that melanized wild-type conidia can scavenge calcium ions. And they may be very important as signaling, as messengers to activate, for instance, calmodulin and signal transduction pathways, which are important for the formation of these lipid rafts. Okay, now I would like to switch to the second part, which is a bit shorter of my talk. And this is, I would like to concentrate on neutrophilic granulocytes. Because what we know is, and I would just describe the identification of a novel killing factor, which is antifungal extracellular vesicles. And what we know is that these neutrophils are absolutely essential for defense against Aspergillus Fumigators. They can phagocytose conidia, of course, also they can degranulate, and also my group and also the group of Frank Ebel showed that they can produce neutrophil extracellular traps against Aspergillus Fumigators, but they do not kill. So the question is, how do neutrophils extracellularly inhibit or kill Aspergillus Fumigators? And when we further analyze this question, we bumped into the production of extracellular vesicles. Just to remind you, I think all cells, maybe it's not sure, but I think all cells on Earth produce extracellular vesicles. When we think about neutrophils, and this is true also for maybe all cells, they produce exosomes, they come from multivesicular bodies, and they produce microvesicles with sizes above 200 nanometers. Also apoptotic bodies and apoptotic microvesicles, there's methods to distinguish all of them. Now, when you have neutrophils, they spontaneously produce a number of vesicles that you can see here by a nanotrack analyzer. And when we infected these neutrophils with Aspergillus Fumigatus conidia, what you can see here, there's an increased production, at least of this distinct type of extracellular vesicles compared to the spontaneously released vesicles. And when we analyzed them further, we found these vesicles here have an antifungal activity. And also by differential light scattering, we found out how the composition of these vesicles produced by neutrophils is. They have different sizes and different percentages. Now, when we isolate these antifungal EVs and we add them to Aspergillus fumigatus, we can see, and this is here in the lower panel, you can see that the growth of these hyphae, and this is here, the germlings at hyphae, is reduced compared to untreated controls. And also what is really interesting to mention that they are located in a different size in fungi. Here we use the fungus that is labeled with GFP, the cytoplasm, and the cell wall with chalcofluor white. And here's the diameter of this fungus. Now, what we see first is that we see really these vesicles also entering the cytoplasm here, clearly in the cytoplasm. Beautiful animations produced by the group of Thilo Figge in our institute. Then we find vesicles that are in their interface, can be found between cytoplasm and cell wall, shown here. And we find vesicles that are in the cell wall, here. And we find, of course, vesicles outside, the majority, shown here. And we can count them. Majority is outside, 50 to 70% in the cell wall embedded, 0.5 to 2.5%, cytoplasmic, but also intracytoplasmic. Quite a number can enter really the cytoplasm. So the question is, can we also measure killing of hyphae in situ? And the answer is yes. We use the strain produced by Johannes Wagner's group, which has labeled mitochondria. And when the hyphae are healthy, they have this tubular structure. And if they are dead, they are clustered and fragmented. And when you use this strain, healthy, nice tubular structure, H2O2 fragmentation, spontaneous vesicles, nice, healthy, long mitochondria, antifungal extracellular vesicles fragmentation. So really these vesicles kill fumigators. Now the question is, what is the potential killing agent of these vesicles? And for that, we did a proteome analysis of these vesicles and either spontaneously produced one or induced with Aspergillus fumigatus. And what we found is we found an enrichment in the antifungal extracellular vesicles of antimicrobial peptides, like cathepsin G or surocidin. And now the question is, how can we prove that they are important? And for that, we expressed these human genes in Aspergillus fumigatus using a promoter system, TET-on promoter system. And when we did that, we killed Aspergillus fumigatus. And for example, here is asurocidin, the gene system is silent. And when you add doxycycline, induce the expression of asurocidin, there is no growth anymore. And you can quantify that when you induce production of asurocidin and cathepsin G intracellularly, no growth anymore. So if these vesicles transport these peptides into the cytoplasm of fungi, they can be killed. So what I showed you is that the EVs decrease the growth of Aspergillus fumigatus. They can even enter hyphae and they can kill hyphae. It doesn't mean that they have to enter hyphae to kill hyphae, of course. My last point, the problem with neutrophils is they are very difficult to work with. They have a half-life time of 18 hours. So is there any hope for a cell line? And I think there is. And this, we were very much motivated by work of Arturo Zieliński's lab in Berlin, Solberger et al. And they showed this cell line can produce neutrophil extracellular traps. And what we found is that these cells shown here, when they were differentiated, they can phagocytose conidia. And also we see the same phenotype, more acidification of conidia and phagosomes, compared to Conidia, wild-type Conidia. And also, if you can quantify that, it's very similar to that what I showed you before. And most importantly is, can they kill hyphae of Aspergillus fumigatus when they produce extracellular vesicles? And the answer is yes, they can. This is primary human neutrophils. This is these differentiated cells from the cell line, untreated, nice tubular structure of the mitochondria of Fumigatus, H2O2, of course, nothing left really. Spontaneous extracellular vesicles, still tubular structures from primary neutrophils, a bit fragmentation, but still broad fluorescence. And if you have antifungal extracellular vesicles, also from the cell line, killing. And this, of course, is really good news, because now we can, if I go back, we can use this as a genetic system to analyze how our vesicles produce and what is their content. So we added another strategy how neutrophils kill Aspergillus fumigatus by producing antifungal extracellular vesicles, a long distance killing. Summary, I spoke about immune evasion, how Fumigatus can interfere with maturation of phagosomes, and the discovery of a novel killing factor. In turn, this is antifungal extracellular vesicles. At the end, let me thank my group. I'm really privileged to have really excellent postdocs, PhD students, master, bachelor students, technicians. We have support in particular from the Deutsche Forschungsgemeinschaft, the Collaborative Research Center Transregio Funginet. And of course, this work was with a lot of collaboration of other groups. In particular, I would like to thank Matt Blanco and his group, and also many international collaborators. Thank you very much for your attention. Oops. Oh.
Okay, thank you very much for a fascinating talk, Axel. So can I remind everybody, please, that to pose a question to our speakers, please put your question in the Q&A box and state the name of the person that you would like to whom you're addressing your question. So it's now my pleasure to introduce our second speaker, Ashraf Ibrahim. Ashraf is a professor of medicine at the David Geffen School of Medicine at UCLA, California, and is based in the Division of Infectious Diseases at Harbor UCLA Medical Center. His early training was in biochemistry and microbiology in Kuwait, and he obtained his PhD in microbial physiology in the UK. From there, he went to work on infectious diseases with Jack Edwards at the UCLA Medical Center, and his research focuses on 3 areas. These are host pathogen interactions, diagnostics and therapies in mucormycosis, vaccine strategies targeting multi-drug resistant Candida species and gram-negative bacteria, and mechanisms of microbial sepsis for development of novel immunotherapies. So we warmly welcome Ashraf as a speaker at MycoTalks, and I'll hand over to him for his talk, which is entitled Unique Invasins and Toxins Promoting Hallmarks of Mucormycosis. Thank you, Ashraf.
Thank you, Alex, for the kind introduction, and it is a pleasure and honor as well to be present with you today in this fantastic forum. So today what I'm going to talk to you about is pathogenesis of mucormycosis as it relates to its angioinvasiveness and toxin production as well. Just here. So as some of you know, mucormycosis is a life-threatening infection that usually happens in the immunosuppressed patients, mainly caused by fungi belonging to Mucorales, and the most common cause of infection is Rhizomas species. As I said, it's basically mainly in immunocompromised patients. It can happen in immunocompetent patients if it gains direct access into the bloodstream, mainly through trauma. Usually, the problem with mucormycosis is the fact that it has a very high mortality, with an overall mortality rate of approximately 50%, and it can approach 100% in disseminated disease, prolonged neutropenia, as well as in cerebral involvement. This is really basically mediated, the lethality is mediated by lack of rapid diagnostics assay. Most of the diagnosis happens through getting biopsies of specimens and tissues. At that time, it's basically pretty much late because of the fact that also the disease is associated with a lot of necrosis, as you can see here from the pictures on the right. These are pictures from immunocompetent patients who were hit with contaminated splinters during the Joplin tornado in Missouri in 2011. Recently, obviously, the disease has been highlighted in the media because of the fact that COVID-19 associated mucormycosis became a major problem, especially in India, with approximately 48,000 cases reported within a very short period of time in the summer of 2021, 2.5 months. These are usually happening in diabetic patients who have been treated with large doses of steroids, which obviously worsens the diabetes as well. So the major manifestation of the disease is rhino-orbital cerebral mucormycosis, as you can see here, and also pulmonary mucormycosis, both acquired by inhalation of spores. Spores of Mucorales are very common in the nature, pretty much like Aspergillus, although the spore size is probably 5-fold higher. And this is another very common manifestation is the cutaneous mucormycosis. Any infection is very prone to rapidly progress into a disseminated disease because of the angioinvasiveness of, or the nature of the angioinvasiveness of Mucorales in terms of invading host tissue. So as I mentioned earlier, this is a classic example of a Rhizomas Mycelia. You'll see here the sporangiophore carrying the sporangium. Sporangium is full of zygospores or sporangial spores, as you would like. If you actually culture these spores, they start swelling within a couple of hours, and then they start germinating within 4 hours. If you leave them for a couple of more hours, you'll have basically full-blown mycelium. This was shown by Sushan Lee and Joe Heitman to be controlled mainly by the calcineurin pathway, especially in aerated conditions as well. We were interested in studying how does the organism invade tissues as it relates to nasal, alveolar epithelial cells and endothelial cells. And the reason for it is in diabetic ketoacidosis, the main manifestation is basically rhino-orbital disease, whereas in neutropenic patients, the pulmonary disease is the most common. And it can actually, in both cases, disseminate into other tissues as well. And we wanted to understand if there are certain factors in the host, such as hyperglycemia, acidosis, and elevated available serum iron, can modulate these interactions vis-a-vis the receptors on the mammalian cells and the fungal ligands in a way that it basically promotes invasion and dissemination. So what we did is we first conducted experiments to see if there's any difference in the ability of Rhizomas to invade and damage host cells. You can see in nasal cells here, these are epithelial cells. You get basically very rapid invasion within 6 hours, and it actually translates later on into damage to these cells, starting at 24 hours and reaching almost a maximum damage within 48 hours. You compare it to alveolar epithelial cells. These are A549 cells. You'll see the same pattern of invasion, but there is a delayed damage to these cells within approximately 48 hours. You get only 50% of the cells damaged. And you look at endothelial cells, if you put different concentrations of Rhizomas spores, they actually invade endothelial cells really well. This is seems to be host receptor mediated process because different concentrations basically result in higher invasion, but then after that it plateaus. Endothelial cells seems to be actually more susceptible to infection or to damage compared to the alveolar and nasal epithelial cells. So we used affinity purification process whereby we biotinylated membrane proteins of the host cells, and then we incubated them with germinated spores or spores of Mucorales, and we washed extensively to get rid of unbound spores, unbound membrane proteins. And then we eluted them with urea on Western blots, and you can see here in terms of Rhizomas species as well as Mucor and Cunninghamella epiphytomyces, all of them, they bound a 78 kilodalton band on nasal cells. And when we sequenced it, it basically came out to be glucose-regulated protein. So by using antibodies commercially available against GRP78, which is a heat shock protein, you can actually block the ability of Rhizomas to cause damage to these cells. We did the same thing with the alveolar cells. You'll see that actually all tested Mucorales, they bound 130 kilodalton. This was to be identified as an integrin beta-1 alpha-3. And again, with antibodies commercially available against beta-1 integrin, you can actually also block the ability of Rhizomas to cause damage to A549 cells. And in the endothelial cells, we found that the receptor seems to be actually also glucose-regulated protein. As I mentioned earlier, this is a heat shock protein. Under stress conditions, usually gets relocalized to the cell membrane and has been reported to be actually a receptor for several fungi, several viruses in particular, and lately actually has been reported to be a co-receptor for COVID-19 with the ACE2. So as I mentioned earlier, we wanted to study the presence of certain host factors such as hyperglycemia, ketoacidosis. These two factors, they affect the ability of transferrin to chelate iron by a proton-mediated mechanism, and as a result, you end up having also elevated available serum iron in these hosts. So what we did is we used physiologically relevant concentrations of glucose, incubated cells, these are nasal cells, with these concentrations, and we studied the expression of GRP78. And you can see here that there is an elevated concentration or expression of the gene as you go with higher concentration of glucose representing hyperglycemia. This also translates into an enhanced Rhizomas-mediated damage to these cells. And the same thing with ketoacidosis, even to a larger extent, 6-fold increase in GRP78 expression on these nasal cells, again, translates into higher damage to these cells by Rhizomas. And again, elevated available serum iron does the same thing. So it seems that these host factors affect the expression of GRP78 on nasal epithelial cells, and that basically promotes enhanced invasion and damage to these cells. And we studied the same factors, the effect of these factors on alveolar epithelial cells, and we did not see any effect on integrin beta-1 alpha-3. So the question that arises usually when you identify a receptor on a mammalian cell, what's the fungal ligand that actually binds to it? So we've used far western blotting using supernatants from a protoplast, regenerated protoplast of Rhizomas. Usually if you concentrate these supernatants, run them on a non-denaturing gel, and you come with the receptor, in this case in the nasal cells, GRP78, you actually, as a bait, and you use the antibody against GRP78, you can identify a band, sequence that band, and you can actually figure out what the fungal ligand is. And in this case, we found it to be COTH3 protein. We've done the same thing with alveolar epithelial cells, and we found it to be COTH7 protein, and we did the same thing with endothelial cells, and it was COTH3 protein. So what are COTHs? COTH proteins are cell surface proteins that are universally present in all Mucorales that we've sequenced in collaboration project with Vinnie Brunos at University of Maryland. See here that these are all clinical isolates that we've obtained from different sources. And you can see all of them, they have COTH proteins, anywhere between 1 to 7 copies. not only they are present, but they are among the most highly expressed in these isolates, especially in Rhizomas in particular. And we've done other organisms as well. So cut itch, we've taken basically the cut itch proteins, and we've done three-dimensional structural homology as it relates to its binding to GRP78, and we've identified an antibody or a 16 peptide MER. We've taken that and we've raised antibodies against that peptide, and we wanted to study if those peptides basically, or those antibodies, can block the ability of Rhizomas to adhere, invade, and cause damage to the different mammalian cells that we are studying. And you can see here clearly in nasal epithelial cells that the antibodies block the ability of Rhizomas to invade host cells in nasal cells without really affecting adhesion, which is also a phenomenon reported in several pathogens as well, because of the fact that these are different processes mediated by different receptors. And you also see that the antibody was almost completely abrogated the ability of Rhizomas to cause damage to nasal epithelial cells. You do the same thing for alveolar cells, same thing, the antibody was actually blocking invasion, a little bit of the adhesion as well, but it had no effect on the ability of organisms to cause or to reduce damage of Rhizomas to alveolar epithelial cells. And we think this is probably related because of the toxins mediated. Even if you block the invasion, you can actually still secrete toxins and you can actually cause damage, especially if you leave them for a long period of time. Remember, the alveolar epithelial cells, it takes 48 hours to actually cause damage to those cells. Similarly, in the endothelial cell, almost complete blocking of invasion translates to approximately 50% reduction in Rhizomas-mediated damage to human umbilical vein endothelial cells. So with these results, we were actually encouraged to actually study if these antibodies can be therapeutic. So what we did is we infected diabetic mice with Rhizomas intratracheally, and 48 hours later, we've treated them with a single dose of 30 microgram of the antibody, which is determined by pilot studies. You can see that the antibody by itself actually is protective. Approximately 50% of the mice survived this severe infection. Posaconazole had really no effect, and this is consistent with the fact that posaconazole is used in salvage therapy rather as a primary or a first-line therapy. And if you combine these two together, they look like dynamite. You had 100% protection, which is was really pleasantly surprising to us. And we've repeated this actually also with liposomal amphotericin B, and you pretty much have the same thing, 100% protection. And remember, as I said, these are basically given 48 hours post-infection. So these are really established severe models here. So obviously this is giving us hope that we can one day present the first immunotherapy against mucormycosis. And now we currently have humanized antibodies are in manufacturing, showing similar pattern of protection in preclinical models. So to summarize this part of the talk, we basically now understand why diabetics in ketoacidosis are usually infected with rhino-orbital cerebral mucormycosis. What we believe is a susceptible host in diabetic ketoacidosis inhales spores of Rhizospus delamar. They basically encounter first the nasal epithelial cells. These epithelial cells have an increased expression of GRP78 on the cell surface because of the hyperglycemia. In certain cases, acidosis. Both of them, they result in elevated available serum iron. And as a result, the organism is stuck in the sinuses, proliferate with the presence of nutrients as well as iron, cause infection and cause necrosis. If these factors are not present in neutropenic mice, what happens in this case, that some of these spores, they go into the alveoli and cause pulmonary infection. We know in both cases that these organisms can actually evade host defenses, with the results coming from George Chamelos' group, diligently showing that actually Rhizospus can evade host killing by macrophages. And this is basically done through a phagosome maturation arrest, mainly happening by the presence of melanin, as shown by Axel earlier today. And usually this results in inhibition of LC3 associated phagocytosis. What we don't know yet is why do we see all this basically extensive amount of necrosis, as you can see here. And we thought that this is probably related to the production of toxins. This assumption was actually also fueled by the fact that if we take killed hyphae, either by heat killing or even by glutaraldehyde fixation, you could still see that there are actually extensive amounts of production of damage to host cells, including A549 cells and HUVEX as well. And what we did is we actually wanted to see are these factors related to spores or hyphae. And you can see here clearly that if you use spores and you kill them by heat killing, you abrogate the ability of the mixture here or hyphae to cause damage to alveolar epithelial cells. So clearly this is a hyphae associated substance. And through Sameh Sulaiman's work, when he was a postdoc in my lab, he's a natural chemist, so he have done a lot of work basically on trying to purify the substance from the extract by using size exclusion chromatography, as well as three-dimensional chromatographic fractionations coupled with HPLC and analytical chemistry. We were able to actually identify a pure fraction that contained 17 kilodalton, approximately 17 kilodalton protein. And you can see here, if you inject neutropenic mice with this substance every other day for 3 times, you actually induce death. And as a matter of fact, if you inject these mice with the substance within 10 to 20 minutes, you get basically what we call circulatory hypovolemic shock, characterized by the mice basically start breathing fast and shallow. And if you touch them, they are extremely cold. They recover, but then later on, they actually die if you keep injecting them with the substance. You look at lungs taken from these mice, and you'll see that there are serious abnormalities, including extensive hemorrhage, as you can see here, and also the presence of abnormal megakaryocytes in the lungs as well. So in a nutshell, this is really a toxic substance, not necessarily just from a single injection, but repeated injections as well. So we studied the expression of the ORF that we've isolated, and we found that it is basically expressed as the cells germinate. Remember, we said that germination happens by 4 hours and it becomes really extensive in 5 to 6 hours. And we raised antibodies against this pure substance in rabbits, and we used these polyclonal antibodies to study the expression of the protein on the surface of the germinated hyphae or spores. You see clearly here that there is really no expression of the protein. You get expression at 4 hours, and then you get extensive expression at 12 hours, not only on the hyphae, but also in the surrounding as well. So it seems that this is really a cell surface substance secreted mainly on the cell surface, but there's some shedding to the surroundings as well. So Clara Balding, who came to us from Axel's lab as a postdoc, she worked on trying to understand the contribution of this putative toxin to the pathogenesis of the disease. So she knocked down the expression by using RNAi, and you can clearly hear that she was successful in reducing it by 80 to 90%, both at the transcription level and the translation level. You have expression in the empty plasmid, both on germinated spores and much larger spores. And you see basically there is barely any expression here when the mutant was produced. That mutant had defect in its ability to cause damage to A549 cells, approximately 40% reduction. And also the antibody itself basically mirrors what we see in the knockout strain. So you can block the wild type mediated damage by using these antibodies. In the diabetic mouse model, if you inject mice with the mutant and you compare its virulence to the empty plasmid wild type, you'll see here that there is a very attenuated virulence, approximately 70% survival in the mutant versus 10% in the knockout. And also, if you infect mice with the wild type, treat them 24 hours with a single dose of 100 microgram of the polyclonal antibodies raised against Rhizomas, you'll see clear, actually, protection in the treated arm. We also wanted to see if this reduction in or contribution of the toxin to virulence translates into enhancement of damage to the tissues in the lungs. These are all infected intratracheally. We used EPOP-TAC kit, commercially available, and with help from Mark Swithergall. You can see here that mice or lungs taken from mice infected with the empty plasmid have actually very extensive amount of damage in their lungs, as indicated with these brown areas. You quantify them and you compare them to the lungs taken from mice infected with the RNAi toxin, you see basically a clear difference in these areas. And the same thing for those mice that have been treated with the antitoxin antibody compared to the isotype matched antibody. Again, there is actually extensive amount of damage in these mice that have been infected with the wild type and treated with the isotype matched antibody versus those treated with the antitoxin antibody. We've had the luxury and pleasure of collaborating with George Hamelos on this, who has actually human specimens from humans who are infected with mucormycosis versus aspergillosis. We wanted to see whether this is really expressed on human specimens. So we've done immunohistochemistry with the antibodies that we have in hand. You can see here in lungs from Mucorales, there's a lot of expression of the toxin in the tissues itself. And if you look closely here at where it is expressed, these are hyphae stained with calcifluor white. They are basically broad acetate hyphae, very thick. And you'll see that the toxin is basically present on the hyphae, but also is present in the surroundings as well, indicating that this is also shedded in addition to the fact that it is expressed on the cell surface. And there is really none of this present in lungs taken from aspergillosis patients. So we wanted to look a little bit closer on this toxin to understand its mechanism of action. So we've done bioinformatics, and every time we've done this, all the stuff that we basically came up popping are either ricin-like toxin, chain B, or ricin-like toxin. And we've aligned the sequence with ricin, which is, as you know, is a plant toxin, very potent. You can see that the toxin that we are dealing with in our Mucorales has similar features to ricin, including the presence of ricin chain A, which is containing the catalytic subunit that results in prevention of protein synthesis. And this is done by ribosome inactivating protein activity. And also, in addition to that, there is the ricin chain B here in blue. And there are two of them is basically in ricin that you can look at. And obviously, we have a very good homology between our toxin and the ricin B chain in particular. In addition to the fact of this sequence homology, we also would like to highlight that this green brackets here shows the catalytic subunit where we believe it's actually responsible for the ribosome inactivating activity, especially the glutamic acid here is separated from the arginine R by 2 amino acids. So both of them seems to be actually present in that particular thing. I should highlight the fact that our toxin is really not containing two peptides or two proteins connected into together into disulfide bonds. It's really a single peptide that's 17 kilodalton in size. And that's where we think it is basically a type 1 RIP, or what we call type 1 ribosomal inactivating protein, versus the ricin, which is a type 2 inactivating protein. By also doing a little bit of more gene ontology studies, we also, in addition to these sequences similarities that we see, we also have several identities, especially the presence of sugar binding motifs, lectin receptor binding motifs. Also, the N-glycosylase activity is predicted to be present in both, the hydrolase activity present in both as well. I also should mention, in addition to these sequences, there is something called the Ldv motif, which is responsible to cause vascular permeability in ricin, both in animal models and humans, and this is also conserved in our toxin as well. If we've done actually three-dimensional structural homology, and we've searched the Swiss library, and you can see clearly that there is very good superimposable structures between our toxin in red and either chain B, the first one or the second one, with very high TM alliance of basically 0.8 to 0.78. So with these structural homologies, we asked the question, is there any cross recognition or cross reactivity between antibodies raised against these toxins? So we've taken the antibody that we've raised against our toxin, and we studied if it can bind to either ricin and the recombinant protein that we actually produce in Saccharomyces cerevisiae. And you clearly see here that plates coated with our toxin, the antibodies recognize it really nicely in a dose-dependent manner. Also, if you study the plates coated with ricin, you see the same thing to a lesser extent, of course, and none of the isotype matched antibodies that we've used basically bound to these two plates. On a Western blot, our antibodies raised against the ricin toxin can recognize the protein that we've used to raise these antibodies against. And also they can actually also recognize both chains of ricin as well. The question, of course, is, I mean, this is all good. Does this basically translate into functionality? Do we think that our toxin acts in a similar fashion as ricin by inhibiting ribosomes and protein synthesis? So to answer this question, we use the classic cell-free rabbit reticulocyte assay. In the presence of ricin, you clearly see a very potent activity of approximately IC50 of 2.2 times 10 to the minus 11. This is 2.2 picomolar, which is really, really very small concentration. So you can actually irreversibly block protein synthesis. If you do the same assay in the presence of the Rhizomas toxin that we have, you see a similar pattern, and you can actually prevent protein synthesis. But the IC50 that's required to do that was much higher, 800 times or 800-fold higher than what is required in ricin, with 1.7 times 10 to the minus 8. Protein inhibition is all good, but the question is, does it really do this with N-glycosylase activity? In other words, does it really cleave adenine from the alpha-sarsen ricin loop? So in an assay where we took RNA extracted from A549 cells, incubated those cells with the Rhizomas toxin, you can clearly see that adenine is released as you compare it to the standard here. And if you do this, if you can actually break the bond between the adenine and the ribose, resulting in depurination, what you can do later on is you can take that extract and expose it to acetic aniline, and you can actually generate a diagnostic band. It's 450 base pair size, that it tells you that the adenine definitely has been released from that linkage. So in that assay, this is using the cell-free reticulocyte assay, incubated with ovalbumin as a control, ricin or Rhizomas toxin. You can clearly see that you can release that diagnostic band when you use ricin at 1 nanomolar, very low concentration. You can do the same thing with the rice toxin, but at a much higher concentration of 10 micromolar. So just to summarize, we've identified and characterized a cell surface highly associated toxin in mucorales. And I should say that this is present in all mucorales. The toxin damages hosts in vitro and is required for pathogenesis. And the toxin seems to actually suppression of the toxin increases or attenuates virulence. And the toxin has very similarity, structural as well as functional similarities with ricin-like toxins, which are basically well described in bacteria and plants as well. So because of these similarities, functionality as well as structural similarities, we named it mucorricin. And this is probably the first report for ricin-like toxin to be present in fungi and outside the plant and the bacterial kingdom. I would like to basically finish by thanking my group, which basically are listed here in blue alphabetically. And I also would like to mention the role that Sameh did in identifying the toxin, Clara and Mingfu as well, and both previously to studying the toxin virulence as well as studying basically host receptors interaction as well. George has done a lot of work with the cut itches, and EU and Shakti did a lot of work on the structural homology as well as the cross-reactivity. Special mention, of course, is to Elaine Beteta, who is the guru of toxins and ricin in particular. She's the one who developed the ricin vaccine. George Hamelos and Vinny Bruno for years of collaborations as well. And I'll be happy to stop here and take any questions. Thank you.
Thank you very much, Ashraf. We'll now start our question and answer period. So please post your questions in the Q&A session, and using the Q&A tab in the Zoom, not the chat. And remember to name the speaker you wish to address. So we will start with questions for Axel. So Axel, there's been quite a few questions that have already come in for you. Mm-hmm. The first one from Shannon Esther Righi, who asks, can you block acidification of PKSP containing phagolysosomes with exogenous DHN melanin? And also a related question from Xavier Sierra, have you tried to use the Ayg-1 mutant in your models? Because this mutant is not totally white like the PKSP mutant, but also has a strong phenotype.
Yeah, thank you very much for the question. First of all, to the first part of the question, when you use just melanin ghosts, how we call it, so you isolate conidia and then you cook them at high temperature with acids, then you just end up with the DHN melanin. And if you take these particles, they behave exactly like, at least what we found in all parameters we measured, like conidia, like wild-type conidia. So it's really related to the DHN melanin. What we have not done this experiment rigorously. So to have in one phagolysosome, both conidia, wild-type and PKSP, we have not done that. But I believe we also did experiments where we put melanin on beads, and if you have coated beads with melanin, they also behave like wild-type conidia. So at the end, it's really the melanin. Sorry, Julie, I forgot the second part.
The second question was about the Ayg-1 mutant in your model?
Yeah, we checked all these mutants in the pathway, and the strongest effect we saw really is when it's fully melanized, so the full polymer at the end. I know the Ayg, we have all these mutants and analyzed them and produced them in part. But the effect is not as drastic like with the full DHN melanin.
Okay, and I'll ask you one other set of questions, and then Alex will come in. The second set of two questions that I've combined is first from Sandeep Velanki. Have you treated Fumigatus biofilms with purified neutrophil extracellular vesicles from Aspergillus? And have you looked from Kelly Shepherdson, have you looked to see if there are differences in extracellular vesicle production from neutrophils from immunosuppressed patients or from neutrophils treated with corticosteroids, or if the contents of these EVs from the neutrophils are altered? So a lot about the
Really good question. So first part, we did not really treat yet, or we didn't check extracellular vesicles on biofilms. We ask questions at the moment like how specific are these extracellular vesicles only for Aspergillus or for others and so on. We have not done that yet, but remember, this is why I also tried to emphasize on this cell culture model. To isolate extracellular vesicles from neutrophils, you need a lot of blood. And of course, we do that a lot, but it's really laborious. You know, for instance, for our proteomic studies, we needed 2 liters or something, you know, of course, from healthy donors and so on. So we are just doing that, but not really on biofilms, I must admit. The second question is, of course, the most urgent question we are following at the moment. We isolate EVs from immunosuppressed patients and, for instance, under treatment with corticosteroids and see what is the effect there. We are also trying to we have some donors with CGD patients, of course, because our model is really, they lack really mature, functional, active extracellular vesicles. This is what we are just testing at the moment. Also, we use animal models, of course. But I cannot give you any data yet. We also will do proteomics and see what is the difference. But maybe just the last point. I mean, what I showed you that there's enrichment of antimicrobial peptides. I mean, this is only, let's say, a proof of concept. We are not sure whether it's really azurosidine at the end or cathepsin G. What we showed is if it's in hyphae, it will kill. But it could be also a mixture, maybe even microRNAs in addition or whatever. You know, we are just still analyzing that because it's not easy with the system. It took us quite some while to establish all. But the questions you asked, absolutely very, very interesting. And I think it's a very hot field, extracellular vesicles, in all types of diseases for all cells and for everything.
Right. It's one of the hot topics for sure. So Alex, do you want to take over a little bit?
So Ashraf, I have a question from Ted White, who says very interesting talk. And so he asks, concerning the toxin gene, is a similar gene found in other fungi, human pathogens or plant pathogens? And is there a possibility of gene transfer from plants to fungi or vice versa on a plant?
Yeah, well, this is really a very good question. And yes, there are actually genes present in other fungi. There's no doubt about it, but they are mainly non-pathogenic, at least to humans that I've seen. A lot of them are pathogenic to plants, interestingly speaking. And is there a possibility of transfer of genes from fungi to plants or vice versa? Yes, absolutely. I mean, there is always a possibility of that happening. I don't have any proof of it, but it's probably these fungi are interacting with plants at all the time, as you know, present in environment in abundance. So we know that there are actually several fungi that contain the ricin-like toxin proteins as well, especially those with structural similarities to chain B. So they are present in other fungi.
Okay, so I have a question as well about, just to follow on from that, is that have you looked at the sort of phylogenetic tree for all these sort of lookalikes? Because you know, in the environment, maybe you could trace that phylogenetically over an evolutionary period to look at that more deeply.
We haven't, not necessarily deeply in terms of evolution. It's a good point, and maybe perhaps we should go back and look at it. As I said, you know, what we've seen is these ricin-like toxins, especially the ones that for type 1, they are abundant in other fungi, but none of them were basically of interest in the sense of pathogenesis to humans. These were plant pathogens in particular. But you raise a very good point, and I think maybe perhaps we should go back and look a little bit deeply into basically the evolution of this toxin. and how does it go from fungi that are pathogenic to plants, or plants as well as rhizome, of course, and it goes into fungi, and why, how are they as it relates to their virulence in humans as well. So probably this is something we should look at a little bit more deep.
Yeah, okay, thank you very much. And back to Judy.
Okay, so we have an interesting question from Al Brown, who thanks you for a fascinating talk and asks about more of the sort of an evolutionary question. Do you think that the DHN melanin mediated inhibition of phagolysosomal maturation evolved in the context of amoebic predation? With this in mind, does DHN melanin promote the survival of Aspergillus during amoebic predation?
Al, thank you very much for this interesting question. We have published 2015. We started to work with amoeba as well, which has been continued now by Falk Hillmann, who has his own group. And what we found is that it behaves, I mean, we only analyzed in my lab Dictyostelium discoideum, so that might not be the perfect amoeba, I must say. There's so many out there, but what we saw there is very similar to that what I reported to you with macrophages. When they are melanized, they are less recognized. They survive in phagolysosomes, but this is also they produce another toxin. Just to say before I come back to the question, glyotoxin, and glyotoxin kills then even amoeba. Amoeba cannot deal with Aspergillus Fumigators, at least Dictyostelium, others can. And the group of Falk Hillmann showed that they also inhibit the maturation of mature, I don't know, it's called a bit different, of phagosomes in amoeba. But they rupture these amoeba. The DHN melanin is rupturing the phagosomes of amoeba. So it is very early in evolution, I believe, in the interaction. It is the importance of melanin, and that melanin survived, as a molecule in nature, I think comes from the environment. And I believe, when I say so, last point, that it also works in macrophages. I mean, this is because they have learned it. I mean, as you know, this is our hypothesis, trained in environment. And then there is in us is the selection. That is, we can kill maybe 990 or 99.9% of all fungi, except of those which have DHN melanin or, of course, other characteristics.
Okay, thank you. So I'll ask you one more. How are neutrophil this is from Johannes Westman, and he wants to understand how neutrophil extracellular vesicles are getting transported across the cell wall and plasma membrane.
Yeah, it's a very good question. The answer is I don't know. No, I think it's just, I mean, I could not believe it when we saw that they're really within the cytoplasm. But remember, I don't know, it was very quick, but if you look for exosomes are produced within other vesicles. So they have double membranes, right? So they have really two membranes, these exosomes, and a model could be, I mean, that they can enter through the cell wall. I think this was also shown maybe by, for instance, by Neil Gold's group and so on, that it's possible for particles to go through the cell wall. And then one model would be they have two membranes. The outer membrane fuses with the cytoplasmic membrane and then releases the vesicle in the cell. But honestly, we don't know yet the mechanism. We are working on that, of course. It's extremely interesting because I've always taught in my classes there is no phagocytosis in fungi. I still believe that there must be. I don't know.
Okay, Alex.
So Ashraf, I have a question from Julian Naglick. He says, you mentioned that the ricin VDV region causes vascular permeability. The mucorricin sequence is LDV. Will this make a difference? And he says he can't remember if you said you mutated this LDV region, and if so, did you reduce that vascular permeability?
So let me start by answering the second part. And the simple question is no, we haven't done that yet. This is something that it's part of our actually grant renewal that we're going to propose to do, and hopefully we'll do soon. So regarding the LDV versus the VDV, it won't make a difference. First of all, a ricin has both LDV and VDV, and I should have basically pointed that out. In the ricin toxin or the mucorricin, we have LDV. There is no VDV, but it has been reported in ricin-like toxins that either sequence is important for vascular permeability. So it's basically an XDY sequence in these motifs important for vascular permeability. So we believe that this is probably going to affect the permeability. What we know is the mucormycosis does affect permeability of endothelial cells in an in vitro assay, whereby you basically split your cells on a membrane and measure the amount of fluorescent dextrin that can move from the upper to the lower chamber. So if you put mucormycosis on these cells, you definitely compromise vascular permeability in that particular model. We haven't done this in vivo yet, but vascular permeability is known to be a feature of mucormycosis. So a lot of patients who are basically seen with mucormycosis have what we call tissue edema, obviously. And this is an indication of compromisation of vascular permeability. And as a result, it happens that you have vascular leak, tissue edema, organ failure, and death. And that's exactly classic features of mucormycosis.
Okay, so I have a question that I want to ask you is, so you've got these two mechanisms where you've shown that if you knock down either of them, you get reduced disease in a mice model and in tissues. So have you managed to tease apart these the different contributions? So for example, you've got different cell types, different tissue types, and you've got different underlying diseases. Can you say whether they both contribute equally on all tissues, or how does that work?
Yeah, so I mean, you just have to keep in mind that usually, I mean, manifestations might happen, and sometimes you have comorbidities. There is no doubt about it. But I mean, you're talking about neutropenic mouse model versus the DKA mouse model. So we've studied more of the role of host factors vis-a-vis the diabetes, basically, or diabetic ketoacidosis. So we know that, for instance, ketoacidosis seems to be a very major effect on disease manifestation, because of the fact that if you take beta hydroxybutyrate as a representative of ketoacidosis, you get way more expression of GRP78 on the nasal cells and on endothelial cells as well, compared to the hyperglycemia. Consistent with the fact that hyperglycemia, although some patients are not acidotic when they come down with mucormycosis and they only have hyperglycemia, the overwhelming majority of those patients are acidotic at the presentation. Less than 50% of them are none, but there are actually, acidosis is really associated with mucormycosis, be it in ketoacidosis or other forms of acidosis. So that seems to play a major role, and we believe that this is really because of the compromisation of the ability of transferrin to chelate ion. So acidosis in particular seems to actually release more ion from transferrin, and we've shown this in ex vivo models using endothelial cells. If you give mice beta hydroxybutyrate, you can completely make them susceptible to mucormycosis, and these are otherwise immunocompetent mice. They don't have diabetes. Just give them BHB, and they succumb to the infection immediately within a few days. You don't see this in aspergillosis. We've done this, but this doesn't seem to be a factor in aspergillosis, but it's really confined to mucormycosis. In neutropenia, there is probably some iron overload. It happens for other reasons, especially if there is transfusions, blood transfusions. This is associated with iron chelation as well, iron overload. And that might also play a role in predisposing them to more severe form of infection, and might actually also contribute to the worst outcome of the disease in neutropenia, because we know that, for instance, for many mucormycosis, you get 70% mortality, compared to probably in diabetic ketoacidosis, now it's 40%. It's actually in the Western world, it's even less than that. It's in India, it's worse outcome, probably 40 to 50%. In the Western world, probably we're talking about now 30% with proper diagnosis and treatment. And because obviously you have surgery in rhino-orbital disease, you can intervene with surgery, you can get rid of the necrosis that you actually see. You deliver much better antifungal to the infection. It's harder to do this with lung infections.
Yeah, okay. All right, thank you very much. I'll hand back to Judy.
Yeah, okay, we've run out of time, but we'll send all of the questions to the speakers afterwards. A very warm thank you to both of our speakers and to Alex Brand, my co-chair, and also to the MRC CMM staff who work very hard behind the scenes to keep MycoTalks running smoothly. We look forward to seeing you on January 27th in 2022, when our speakers will be Professor Dr. Ilsa Jacobson from the International Leibniz Research School and Professor Gustavo Goldman from the University of São Paulo in Ribeirão Preto in Brazil. Thank you.
Festive greetings, everyone.
Thank you very much.
Happy holidays.